What are prions and the diseases caused by them? Prions are misfolded proteins in the brain discovered by American biologist Stanley V. Prushner. They have the ability to influence other normal proteins in the brain to fold abnormally. Alzheimer's disease, Creutzfeldt-Jakob disease, etc. are the diseases caused by prions.
Prions, as mentioned earlier, are misfolded proteins. However, the process in which it is capable of finding an alternative stable protein structure still requires greater research, as it is still not very well known. However, many model organisms such as yeast has been taken in to study the formation of prions. Prion formation to occur spontaneously is extremely rare. However, there are many ways to increase the likelihood of prion formation. The repeated expansion of oligopeptides in the N-terminal of the prion is one of them. This is because they can help the prion to be more ordered, which allows it to come back to its alternative stable conformation. From previous in vitro studies, it has been found that these prions created oligomers that were capable of aggregation in other oligomers of its same type, thus leading to the next round of prion formation. Stress provided by the cellular or protein environment can also induce prion formation. High density of misfolded proteins around the normal prion can also induce prion formation too. As mentioned earlier, prions that created oligomers can induce other oligomers to be aggregated as well. This relates back to the infectivity of the prion and how it is able to spread as it converts one prion into a diseased prion and so on. Pre-existing diseased prions which have conformed structures will be denoted by PRPSC as in the diagram. They will spread by converting other normal prion proteins denoted by PRPC, which is commonly found around in the central nervous system. The PRPC now will be the new generation of the PRPSC and the process will start again. The process as to how it works is still very unclear. However, scientists now strongly believe that PRPSC is majorly composed of beta-pleated sheets. Scientists cannot confirm the structure of the PRPSCs as no image has been clear enough to be studied. However, PRPSCs do have special structures which indicate that during this conversion to PRPSCs, a great structural change has occurred in the PRPC protein. Neurodegenerative diseases. There are hereditary diseases that result in long-term fatal issues, often due to damages in the neuronal tissue, specifically the central nervous system. Quite often, these diseases are caused by prions, as mentioned earlier, and this can make them very hard to understand or treat as well. Prions are known to cause fatal damage to the neurons, thus eventually leading to fatal situations of it. But to how the prions cause damage to the neurons depends on the protein structure, as structure leads to function. Prions depend on their neurotoxin property in order to cause damage to neurons, which are quite often long-lasting, as neurons cannot recover. This can then lead to cell death, which then causes one less neuron within that area, and in that trend, several neurons may be affected by the prions. The toxicity of the prions is associated with the induction of prions. There are also certain factors or genes that may increase the prions toxicity too.
Alzheimer's disease, a neurodegenerative disease and the main cause of dementia. One of the most common prion diseases, dementia is a condition of memory loss and difficulty in learning. The prions which cause Alzheimer's disease are beta amyloid and tau. How do they cause neurodegenerative effects? Alzheimer's. Amyloid precursor proteins found in the cell membrane of neurons help in growth and repairing of neurons. Over time, it gets broken down and recycled by alpha and gamma secretase. But when beta secretase breaks down APP instead of alpha, it forms an insoluble monomer called amyloid beta. These monomers bond together to form beta amyloid plaques, which disrupt neuron signaling, causing seriously impaired memory. The damage caused by prions and the lasting impacts on neural health. Neurons are held together by cytoskeleton that are made up of microtubules which transfer nutrients and molecules throughout the neurons. Tau, a special protein, ensures the microtubules don't break. When kinase transfers phosphate group to tau protein, it changes its shape and stops supporting the microtubules. The tau protein breaks up and gets tangled, forming neurofibrillary tangle. Neurofibrillary tangle causes neurons to not signal and function properly, sometimes which causes apoptosis of neurons, causing atrophy of brain. Creutzfeldt-Jakob disease is a rare neurodegenerative disease. There are three main types of CJD: sporadic, which has no precise explanation of its cause; the hereditary CJD, the mutation is passed on by family; the acquired CJD transferred via. How do they cause neurodegenerative effects? CJD. Creutzfeldt-Jakob disease is caused when normal prion protein PRPC is misfolded to diseased prion protein PRPSC. Then the diseased ones trigger the catalysis of the normal PRPC to PRPSC in a chain reaction. Damage caused by prions and their lasting impacts on neural health. The brain shrinks and turns into a sponge-like appearance called spongiform encephalopathy, causes brain atrophy. Conclusion. Prions are misfolded proteins which cause neurodegenerative diseases like Alzheimer's and Creutzfeldt-Jakob disease. There are no specific treatments yet, but researches are being conducted by scientists to know more about prions and to find a cure to its diseases.